Good afternoon, dear students. Today, our story will be about insulin. I explain some key moments about insulin structure, about insulin secretion, about the mechanism of insulin action, and some metabolic effects of insulin. I'll explain. First of all, about the structure of insulin. Insulin is a peptide hormone that's secreted by beta cells of pancreas. According to this scheme, you can find insulin is secreted in beta cells in form of two inactive precursors, preproinsulin and proinsulin. Preproinsulin has four parts, four peptides. First peptide is N-terminal signal peptide, is called L-peptide. Next, B-peptide, C-peptide, and A-peptide. The synthesis of preproinsulin is located on ribosomes of endoplasmic reticulum. Preproinsulin is transformed into proinsulin into Golgi apparatus, and this transformation will be as a result of removal of L-peptide and formation of two disulfide bonds between B and A chain. This is a proinsulin. Proinsulin is transformed into insulin by removal of C-peptide. After removal of C-peptide, we can find here the structure of this peptide hormone, insulin. By the way, concentration of C-peptide can be defined in bloodstream and can give information about synthesis and secretion of insulin. How insulin is secreted? Insulin is secreted if concentration of glucose in blood is increased. This is the scheme of beta cell of pancreas, and here in cell's membrane you can find specific transport proteins. These proteins is called GluT2. This protein is used for glucose transport into cells, and in our organism there is a family of these proteins: GluT1, GluT2, GluT3, and GluT4. The first, second, and third carrier proteins are located in insulin-independent cells. The quantity of these proteins in cells membranes is not dependent on insulin concentration. Concentration of quantity of GluT4 proteins in cells membrane depends on concentration insulin in blood. GluT4 proteins is located in cells membrane of insulin-dependent tissues. Here we can find that for transport of glucose into beta cells, there are special proteins. These proteins are called GluT2. GluT2 is active if concentration of glucose is increased in bloodstream, and in this case there is the formation of glucose gradient. After transport of glucose into beta cells, glucose is phosphorylated and will be involved into metabolism. Glucose is transformed into pyruvate. Pyruvate is transformed into acetyl-CoA. After there is stimulation of TCA cycle working and oxidative phosphorylation, and the final result of this metabolism is increasing quantity of ATP. Concentration of ATP is increased, and this is signal for closing of potassium channels. Potassium channels will be closed, and concentration of potassium in beta cells will be decreased. Increasing the concentration of positive ions leads to depolarization of membrane. Depolarization of membrane opens calcium channels, and concentration of calcium ions in beta cells will be increased. By the way, concentration of calcium channels will be increased because calcium will be transported from endoplasmic reticulum too. Calcium is the key ion for stimulation of insulin secretion from insulin vesicles into bloodstream. And after it, concentration of insulin in blood will be increased, and insulin will do all for decreasing glucose concentration in bloodstream. The main task of insulin is really decreasing glucose concentration. And how insulin can do it? Next moment, I'll explain the mechanism of insulin action. Insulin decreasing glucose concentration because insulin activates two processes. First process is transport glucose and glucose utilization. You should remember two keywords: transport and utilization. Insulin decreases glucose concentration in blood because insulin can stimulate the transport and utilization of glucose. Now I'll explain how insulin can stimulate the transport of glucose into insulin dependence tissues. By the way, here you can find GLUT1 and some words I'll say about insulin independence tissues. Insulin reacts with insulin receptor. This receptor is located in cells membrane because insulin is peptide hormone and cannot penetrate into cytoplasm. Insulin receptor has interesting structure. Insulin receptor has two subunits, alpha subunit and beta subunit. Alpha subunit is necessary for binding insulin with receptor. After binding insulin with receptor, beta subunits will be activated. Beta subunit is enzyme. This is tyrosine kinase. This enzyme phosphorylates proteins and enzymes in cytosol. First protein that will be phosphorylated is IRS protein. IRS protein, after it will activate phosphatidyl inositol 3 kinase. Phosphatidyl inositol 3 kinase will activate protein kinase B. Here you can find AKT. This is another name of this enzyme. And final result of this activity is translocation of vesicles with GLUT4 to cells membrane. And after it, concentration of GLUT4 in cells membrane will be increased. You can find it here. If concentration of insulin will be increased, there is fusion of these vesicles and there is increasing of quantity of receptors, and after it, glucose transport in insulin dependence tissues will be stimulated. If concentration of insulin will be decreased, quantity of these carrier proteins in cells membrane will be decreased because there is internalization of these carrier proteins. You can see here GLUT1. GLUT1, these are proteins for transport of glucose too, but these proteins are located in insulin independence organs, and according to this picture, you can find the activity of this protein depends on glucose gradient concentration. And now insulin can stimulate the transport of glucose into tissues, and it is why glucose concentration in blood will be decreased. In the same time, as I said, insulin stimulates the utilization of glucose in tissues. Some metabolic processes that are activated by insulin are here. For example, here you can find glycogen synthesis will be stimulated and protein synthesis will be stimulated. By about metabolic effects of insulin, I'll explain by using next scheme. And now insulin decreases glucose concentration in blood because insulin activates utilization of glucose. What does mean utilization? Utilization is transformation of glucose into compounds that are not glucose molecules. First of all, by green you can find which processes will be activated by insulin. Glycolysis, glycogen synthesis will be activated because these processes are processes of glucose transformation. From carbohydrates metabolism, insulin activates three processes. First of all, I said glycolysis, glycogen synthesis, glycogenesis, and pentose phosphate pathway or hexose monophosphate shunt. Pentose phosphate pathway that activated by insulin is power source of NADPH. NADPH is used for lipogenesis because insulin in the same time stimulates lipogenesis in adipocytes. Glucose will be uptake by muscles and adipocytes and will be transformed in triacylglycerols, especially in adipocytes. Insulin stimulates the synthesis of free fatty acids that will be used for the synthesis of triacylglycerols. As a power anabolic hormone, insulin stimulates not only lipogenesis, insulin stimulates protein synthesis. Stimulate protein synthesis through activation of transport of amino acids into cells, plus stimulation of transcription. Insulin stimulates the synthesis of proteins too. In the same time, insulin will inhibit processes that are sources of glucose. First of all, gluconeogenesis, glycogenolysis. These two processes are processes for glucose production. Insulin as an anabolic hormone inhibits lipolysis. Insulin inhibits beta oxidation of free fatty acids, and that is why insulin inhibits the production of ketone bodies. If you know, ketone bodies are products of non-full oxidation of free fatty acids. If insulin inhibits beta oxidation, insulin at the same time inhibits the production of ketone bodies. Insulin as an anabolic hormone inhibits proteolysis, and that is why insulin inhibits the degradation of proteins to free amino acids. And now you should remember, insulin is the main hypoglycemic hormone, and insulin decreases glucose concentration in the blood because insulin activates the transport and utilization of glucose. Thank you for your attention. I hope this information will be useful for you.